Hello, everyone, and welcome to today's webinar. I'm Chris Boomsma, Chief Executive Officer here at the Council for Agricultural Science and Technology, or CAST. And it's really the privilege of myself and our organization to partner today with SIVB, the Society for In Vitro Biology, as we jointly offer a webinar on From Code to Culture, How AI and Bioinformatics are Transforming In Vitro Biology. So CAST was formed in 1972. We have 50 years of working on our mission to provide credible, unbiased, science-based information about food and agriculture to policymakers and multiple other audiences. And now a little bit more about CAST. First of all, we're a US-based, internationally connected organization, a 501c3 nonprofit that is based on membership, both organizational and individual memberships. What's unique about CAST is we span the animal, food, and plant domain with regards to science and technology and talking about science and tech. We are nonpartisan, science-focused, and everything we offer in education is free to all. Our mission, as I referenced earlier, is to convene and coordinate networks of experts. You're going to hear from some experts today through our collaboration with SIVB. And those experts are helping assemble, interpret, and communicate credible, unbiased, science-based information. And you can see our audiences there. The first one we focus on is often the one we focus on most, which is policymakers. But organizational leaders, educators, scientists, the media, the private sector, the public, all matter to us and are all audiences of CAST. We can't do our work without the generous support of the many volunteers that give to CAST. They come from academia, nonprofits, government, and companies. They form our work groups, our board of directors, our advisory council, and the like. And so we thank them very, very much. And we thank the volunteers joining today to help put on this webinar. Just a little bit of background about what we're doing here at CAST. We did form a strategic advisory council recently to chart a path to become a National Academy of Agriculture. Why now? Because as you know, agriculture faces rapid technological change, a lot of geopolitical competition, consumer expectations are shifting, and we have an evolving food and national security landscape. So in response to that, we're moving forward, building out this council and considering how to chart a path to become a National Academy of Agriculture. Stay tuned, follow us on social media, and you'll learn more about this endeavor. If you're interested in CAST and getting more involved, look at the QR code right now on your screen. Use your phone to take a look at that, scan it. You can learn more about membership. You can also get involved in our educational endeavors, whether you want to offer us topics, be a contributor or reviewer, help with an event hosting. All of it's fair game, and we'd be happy to interact with you. You can also engage through our newsletter, be part of that. We'd love for you to be part of our social media work. Network with us, get involved in student programs, and support us in a variety of ways, including membership. If you'd like to explore more about CAST in general, just simply take a look at the QR code at the screen right now. Click on that and engage. We'd love to hear from you. By the way, in the lower right corner of the screen at this time, you can reach out to me via LinkedIn through the QR code. And of course, you can reach out via office or cell or email. I'm happy to have a chat with you. And with that, I'm now going to hand it over to Deepika Chauhan, who is the moderator of today's webinar and is chair of the SIVB Membership Committee and has been instrumental in organizing this event on behalf of SIVB. She'll be moderating today's conversation, introducing our speakers, and managing the Q&A. Deepika, thanks so much for joining today. The floor is yours.
Thank you, Chris. On behalf of the Society for In Vitro Biology, we are pleased to welcome everyone to this joint venture of CAST and SIVB. Established in 1946, SIVB is a professional society dedicated to advancing biotechnological research and scientific knowledge at the cellular and developmental level. The society fulfills its mission via 2 journals, online newsletter, annual meetings, workshops, and educational collaborations. Today's webinar supports our mission. The SIVB membership and education committees are working on more webinars, so keep an eye out for webinars this year. If you are a current member who has not yet renewed your membership for 2026, Or if you are not presently an SIVB member but wish to support our mission, we invite you to join the SIVB at members.sivb.org. Our membership is calendar based, so all memberships begin in January. SIVB membership offers a range of benefits such as online access to our publications, discounted meeting rates for sharing your research and networking, volunteer options, and eligibility to our awards. And speaking of meeting registration, you still have few days to register for SIVB's 2026 in vitro biology meeting at the Peppermill Resort and Spa in Reno, Nevada. From June 6 to 10, 2026, the program promises to be an exceptional event with sessions on advances in animal and plant technologies, natural and induced genetic variations, synthetic biology, and use of AI tools and techniques in research, and so much more. Please scan the QR code to the left for more details of the program and on the left to register. We are also thrilled to share that SIVB will be celebrating our 80th anniversary at the meeting with two special keynote presentations on Sunday from Dr. Indira Vasil and Delia Bethge, and a special reunion celebration on Monday, June 8th. I hope you will register and join us. Scan the QR code on the left or visit sivb.org/meetings for more information. In addition, SIVB is pleased to announce the success of our match mentoring program, where postdocs learned about trends in plant and animal biology, improved their leadership and communication skills, and expanded their network. This year, the program is open to both postdocs and students. The deadline to sign up as a mentor or a mentee for 2026 program is today. And all participants must be active SIVB members. If you are interested, you can contact our SIVB business office at sivb@sivb.org or scan the QR code that you see on the screen. Now let's get started. Our first speaker today will be Dr. Hans Bethge. Dr. Bethge holds a PhD in horticultural science from Leibniz University Hanover and University of Applied Sciences Osnabrück, where his doctoral research focused on automated phenotyping in plant tissue culture. He's currently R&D project leader at PhenoLytics GmbH, leading activities in X-ray phenotyping of seeds and seedlings. His research sits at the intersection of plant biology, imaging technologies, and machine learning, with contributions spanning from automated multi-sensor phenotyping systems, machine learning-based detection of physiological disorders, multimodal imaging, and AI-driven optimizations of plant tissue culture media. He is a lead developer of ADAM, an open access platform for data-driven optimizations of biotechnological processes. Following Dr. Bethge, we will welcome Matthew Gruner. He uses a combination of biology, data science, and statistical model to explore high-dimensional relationship between brains, behavior, and longevity. Working alongside Doctors Meet Zandewala and Teresa McKinn at the University of Nevada, Reno, he leverages connectomics and single-cell atlases of arthropods, including fruit flies and ants, to chart the deep evolutionary history of peptidic cells. His research aims to uncover how these networks drive social behavior, metabolisms, and lifespan across the tree of life. After both the presentations are completed, we will offer the opportunity to address any questions you may have. You are welcome to place your questions in the chat during each presentation, and we will share them with the speakers during Q&A session. This webinar is being recorded and will be made publicly available soon. Now, with the introductions out of the way, it is my pleasure to welcome Dr. Hans Bethge, who will be speaking on Beyond Institution, Digital Phenotyping and AI-assisted media design in plant in vitro biology. Welcome, Dr. Bethge.
Thank you very much for the kind introduction. In the next minutes, I would like to talk about how we can use digital phenotyping to quantify plant traits in plant in vitro culture, and how we can use machine learning for culture media design in plant in vitro culture. My presentation is structured as follows. I will start with a short introduction and then continue with describing a robot system that we built for plant tissue culture. Then I will show you how we make use of the data from the robot to automatically detect a certain growth anomaly. In my last part, I want to show you the software that we have developed which can be used for culture media design. So as you all know, plant in vitro culture forms the base of all or most of all biotechnological methods for plant breeding, and it is used for large-scale multiplication of genetically uniform and virus-free plants. However, often there are contaminations, disorder, or endophytic outgrowths occurring. Therefore, this demands a frequent manual inspection and monitoring. Due to high labor cost, there exists an enormous potential for automation. Therefore, we thought in my PhD thesis, a robot that could monitor these plants would make a lot of sense. But what are these traits that we are interested in plant in vitro cultures? So we would for sure like to know the biomass growth, demultiplication rate, and the shoot length, and for sure also the plant quality. Up to date, most of the evaluation methods that we use are destructive and invasive and limited to single endpoint measurements. This could be due to the fact that if you want to apply phenotyping, so imaging, to a plant in vitro culture, you face a highly challenging situation. So you have water condensation on the lid of the vessel. Also, there is scattering effects from the illumination on the lid of the vessel. And even more, there can be mirroring of explants in the vessel walls. So to counteract against these challenges, we use the so-called bottom cooling system to prevent water condensation. We substitute our lid by a plastic foil, a sterile foil, to have a fully transparent view, and we use diffuse illumination to minimize the scattering effects and mirroring effects. Here you see now the hardware of the robot system that we built. As project money was very limited, we used a commercial laser cutting platform. You could also imagine a 3D printer platform. We added a Z-axis to account for different focal planes, added some network components to communicate with the robot, and developed our detection head. Inside this detection head, we have, on the one hand, illumination, like LED illumination and diffuse ring light to illuminate the plants, and 4 sensors installed: an RGB camera, a thermal camera, a point scanning microspectrometer, and a point scanning laser distance sensor. Today, I only have time to talk about the RGB camera. And this whole robot system was built in a space-efficient way that it fits to a multi-layer shelf system where normally in vitro cultures are grown, so that the robot can phenotype these plants while cultivation. Here you see the robot in action, so it sequentially approach the different vessels and does take different measurements. These different measurements are illustrated in the scheme of the data acquisition. Also, due to the limited of time, I will not go into detail, but we phenotype different plant species in different developmental stage. For example, Arabidopsis thaliana as an in vitro germination example, Malus domestica in shoot multiplication phase, and Tabacum for shoot and root regeneration. Every day, 6 images per vessel were acquired, up to 10 to 20 vessels. Then we ended up with an high-resolution image. But if you want to make measurements on these plants, segmentation is needed. And despite the fact that we are already in a controlled environment, like an in vitro lab, there's still a lot of variance inside the data. For example, these sources are coming from different light intensity fluctuation from the tubular fluorescent lamp, or mirroring effects, different media colors, plant species, or explant types. This is not efficiently solvable by an image processing pipeline based on classical methods like color transformation and thresholding or morphological separation. Therefore, we used a machine learning approach with a toolkit called Elastic, where you partially label pixel into different classes. In this case, we only wanted to label into background and foreground. Foreground means plants. And then a random forest model predicting the class membership. By doing so, we labeled partially 50 different images and could then process 1,500 images from the same time series data set. Here you see the pipeline in more detail. So we have our high resolution raw image. Then we do some auto contrast, and we downsize the original image to 1/10 of the original resolution to have less prediction for the random forest model to be more efficient. And the random forest model predict a binary mask, meaning 0 means background, 1 means foreground, plant in this case. We resize this binary mask to original resolution, and could then edit up these 2 data sources together that we have a nicely segmented image. By doing so, we could visualize the in vitro growth of Arabidopsis thaliana, here dancing in the rhythm of life in this culture. On the left side, this is the raw image. On the right-hand side, the predicted or segmented mask. When you have these segmented images, you can do multiple things. For example, you could compare the treatment of different media, for example, as on vessel level, that you sum up the projected plant area or mean average projected plant area per vessel and compare the treatments. Or if leaves are not overlapping, you can separate these individual explants and do measurement of the explant on explant level, like the plant area, or shape features like solidity or compactness. We also monitored the growth of Malus domestica in shoot multiplication. On the left-hand side, you see the raw image. In the middle column, the segmented data, and on the right side, you see the projected plant area. So summing up all plant pixels for this vessel. At the beginning, you also saw the effect of the binaural rhythm more strong. Yeah. And if you have these growth curves, you could also try to interpret these data. Yeah. So for example, we see a smaller growth due to maybe the stress-induced transfer, or yeah, and then we see a linear growth. Could mean that we see cell elongation of already formed leaves. Then we see again a decelerated growth. This could be attributed to the development of axillary shoots, and again a linear growth that these newly formed shoots elongate. In the second part, I want to show you now how we can use these images to automatically detect hyperhidricity, a well-known growth anomaly. So here we have the RGB images. We labeled them with so-called bounding boxes around each explant, and they have two different colors for hyperhydric explants and non-hyperhydric, so normal plants. Then we did again some auto contrast on the images, and more important, we applied augmentation to increase the data set, but also to make the model more robust against fluctuations in light intensity, or applying noise, which also could come from the camera. Afterwards, we have split the data set into train, validation, and the test set, and trained different machine learning models for object detection. And with only 375 images, we could get a high precision and a high recall, meaning most of the explants were found, and the precision means they were also correctly assigned to the correct class, either normal or hyperhydric explant. Now you see the the prediction of the model. On the left side, you see our control group with agar, and on the right hand side, you see a forced media, or a media that forces the occurrence of hyperhidricity with low amount of gel ride. And then you also see the bounding boxes, and the number above the bounding boxes is the probability of the model for assigning a class membership. What you see is on the left-hand side, most of the explant are assigned to be normal, and on the right-hand side, so where we also expected hyperhidricity, the model correctly classified these explants as hyperhydric. In my last part, I want to now go briefly into the software that was published today in Plant Methods. The software is called ADAM, and can be used for culture media design and optimization. So as you all know, culture media is the key factor for efficient micropropagation, but this culture media design is often intuition and experiments-based. Hildebrand et al., for example, needed 16,000 runs, like individual media recipes, to create their basal medium, or Muragashi and Scope spent 5 years in the lab. So this classical culture media design in plant tissue culture is often a trial and error approach, and also OFAT approach, which means only changing one factor at a time. And if we, for example, create experimental design based on a full factorial design for 10 different media components tested on 3 different levels. If we want to have all interactions in our design, that would mean we have to cook 60,000 individual medias, which is not realistic. Also special to plant tissue culture is that we often have multiple goals that we want to maximize or minimize, and they are often conflicting objectives. So, for example, having a high shoot number by also having large shoots. So therefore, we created this browser-based platform called Adam. It's written in R as a R Shiny app, and it covers the complete end-to-end workflow, starting from designing experiment and ending up with identification of an optimal solution. So in the software, we use machine learning for modeling and evolutionary algorithms for optimization. There are no licensing barrier, and supports a reproducible workflow and in-memory operation only, so no data are stored. Here you see the 5 main types of the software, starting with designing an experiment. So you could choose different concepts of design of experiments. For example, Latin hypercube sampling, where you set that you only can realize 50 individual medias in your lab. And once you have created the design, you see the concentrations that you have to test in your lab. And then you have run the experiment and can upload these data. And what the first thing that you have to do is uploading the CSV, where you have the inputs, meaning the culture media components, and the outputs, so the plant responses, for example, shoot number or shoot length. And the software would like to know what kind of variables are there. So what are the inputs, what are the outputs? If the model or if the software knows this, we first want to train or want to know a relationship between inputs and output, and we do this with machine learning. So first thing what we do is splitting the data into training, validation, and test set, and then training different kind of machine learning models. On the bottom left figure, you see now the performance of a trained random forest model predicting the shoot number based on media components and on the performance on the test set. What the software also shows you is the importance of different features. So for example, for this random forest model, predicting the shoot number, the most important input was cytokinin BAP, which does make sense. In the software, we make use of natural selection. So you always start with creating synthetic media components, then use your machine learning to predict the fitness, and only good parents are allowed to recombine, so to give birth to children, and this happens over and over again. At the end, you see the optimized solutions that you then can test in your lab to validate. ADAM have different components. Here is the QR code. So far, 5 concepts of DOI are implemented, 9 machine learning models are there, and 4 different optimization algorithms. We hope that with ADAM, we can transform the tissue culture media optimization from an empirical intuition into a systematic data-driven approach. I will now close my talk with a short video that I will speed up a little bit. And as I'm now working for an X-ray company, maybe also, phenotyping plant tissue culture with X-ray can be a solution. With this, I would like to end my talk and thank you for your attention.
Thank you, Dr. Bethge, for such a fascinating presentation. Now, I would like to welcome Matthew Gruner, who will be speaking on From Synapse to Single Cell, Improving Universal Cell Embedding via High-Resolution Arthropod Connectomics. Welcome, Mr. Gruner.
Sorry for the short delay. So it's my great privilege to talk to you today from Reno, Nevada, near Lake Tahoe. So I'm a team member in Dr. Meet Zandewala's lab. So we're very fortunate to have a multinational lab where we have a lab here and in Louvain, Belgium. So our main interest is in understanding the basic biology of neuropeptides. And this has grown, in huge prominence recently with the incredible usefulness of the GLP-1 antagonists, amongst other things. And we believe that arthropods are a great system in order to understand the basic mechanisms of this. And I'm going to try to make the case for why I think this actually could relate to all other aspects of biology. And the key technology that's enabling this is really high dimensional imaging technologies, like you can see here. So we study Drosophila as our main model system, and we're part of the Flywire consortium that's created the first full brain connectomes of an animal. And now there's multiple connectomes of the fruit fly. They're composed of hundreds of thousands of extremely high resolution images, down to 4 nanometer resolution, that using AI, some techniques I'll talk about a little bit, use advanced segmentation models to piece this all together. We actually work with a private company, Zeta AI, to do this aspect of building a connectome. And it provides unprecedented detail of cellular biology that's occurring in the brain and potentially other tissues. So using this, we can create a lot of information. And actually, that's where we're at now, is trying to understand how to make sense of all this information in the context of all other biology. So the fly connectomes are great and unique in that they're fully accessible to the public right now. You can mine this data, anybody can mine this data if they want, using some incredible packages, R packages from the Natverse system. So yeah, I'll just let the video finish. And using UNet segmentation models, we can actually predict which synapses actually secrete which neuropeptide. So our lab is particularly interested in a different kind of synapse than it's typically studied in neurobiology, perhaps. These would be the ones that secrete neuropeptides. So these don't associate with the same exact positions in neurons that the vesicular synapses do. And so a lot of the original annotations of the connectome didn't include dense core vesicles. So we're actually developing our own model. So it's a little bit of background on the neuroendocrine system. So these are the specialized cells that secrete neuropeptides. In mammals, this would you would be focused on the hypothalamus and the pituitary gland. So these are embedded neurons that, neural regions that are embedded in the central nervous system, but then also secrete and communicate throughout the body. So to orient you to the insect neuroendocrine system, you can see that it's pretty analogous. We have a central nervous system where the nervi corpora cardiaca is the site where neuropeptides are actually secreted into the hemolymph or the equivalent of the blood supply. Where and also neurons that descend from the central nervous system into the ventral nerve cord also secrete into the hemolymph. And these, as you can imagine, play really key roles in regulating metabolism, lifespan, major life transitions in the insect system. So the original studies in insects, because insects have really manageable sized brains, they've been studied as models for understanding the neuroendocrine system in a whole animal for many years. And some of these, a lot of these studies were done in non-model systems like the blowfly, where they would start by doing dye backfilling assays, where they would give a dye to the sensory neurons, perhaps, of the blowfly, and then take fluorescence images of these. And what we found now with this much higher resolution technology really fits, of course, what we found in those studies. And so one of the main focuses of our lab is to understand both the structure and function of neuroendocrine cells in essentially all arthropod systems. So once you have an EM connectome generated, it's really a prediction by the AI. It's not perfect, and it requires a lot of man hours actually to go in and to fill in the fragmented sections that the AI predicted. And so we actually spend a lot of time going through these EM sections manually to actually find the fragments and sort of like a big jigsaw puzzle, put them back together. And our first major study was completing the neuroendocrine connectome in the fruit fly. This occurred about 2 years ago. We're still working on the publication that's coming out in eLife. But with that, we were able to identify all 10 populations that were known in the fly, as well as some unknown populations of neurosecretory cells that we are now interrogating in the lab. So that's pretty exciting. So the morphology is extreme, and it's fascinating. And real honestly, the neuroendocrine cells, they make a number of synapses, but there's other neurons in the fly that make even more, right? That can span the entire brain. So this high dimensional morphological data, I think is showing to be really amazing in what we can discover from it. Even the stuff we haven't, the questions we haven't brought to the system, but the questions it's begging to ask. So once we have created a connectivity map throughout the whole brain, this now exists as in a vector dimensional space. And in this kind of high dimensional vector space, it's actually very easy to find things that have like neurons that have similar numbers of connections or morphologies. All you have to do is take the cosine between the vectors, and often you can get really good segmentation of the different neurons, like you can see here on the left. But neuropeptides are kind of, as a neural signaling molecule, are really interesting because they signal beyond just the brain to other organs throughout the animal. And so tracking these changes requires additional tools, mostly based on fluorescence microscopy. So we're also having a lot of projects in the lab that are focused on using these to map where neuropeptides that are generated in the brain and where they act throughout the animal. So a recent paper spearheaded by a postdoc, Jayati Jera, found the ITP signals, or the ITP neurons in Drosophila for the first time, annotated them for the first time, and was able to find the unknown neuropeptide receptor, a guanylate cyclase, in Drosophila. And people have been looking for many years to find this, and she was able to show that they played an important role in regulating the clock. In fact, they make direct synapses with the clock and are part of the clock system. And they regulate the osmolarity and the retention of moisture in the fly. And they do this by signaling to the gut, and including the adipocytes in the fat body. And so understanding, we're able, with these new connectome methods, we're able to understand how the structural relationship of the neuron, where it's at, can actually relate to essentially all aspects of biology in the fly. So we continue our studies to fully understand the neuroendocrine system in the fly. where you can have a range of sensory inputs, including gustatory, olfactory, and clock inputs, time of day, and including transitions in the environment that are mediated through neuromodulation to change metabolism. And these are very analogous systems actually to mammals in that they're really highly conserved, and a lot of the same metabolic mechanisms are conserved. So I'm also really excited to tell you about an ongoing collaboration with Daniel Cronauer's lab at Rockefeller University, where we're annotating the first ant connectome. So we use the species Uceri biroi, which is a very interesting ant species. It has no queens. Colonies are entirely composed of small blind workers that they go through a process of actually parthenogenesis. So it's all females. And they live exclusively on the live young of other ants. But this actually kind of surprisingly makes it an incredible, I would say, in vitro system, one that you can be grown in the lab and studied, because you can create a clonal line from just one ant. And in fact, they're clones of each other. Because they're clones of each other, their connectomes are unmatched in terms of their similarity. Okay. Yeah, and so this work is ongoing, and to annotate the neurosecretory cell network of the clonal raider ant. So far we found 153 neurons, so slightly more neurons than the fruit fly, and we're actually annotating the presynaptic partners of the neurons to understand all the potential sensory inputs and other kinds of inputs into this neurosecretory network. And we're finding that, structurally there's a lot of similarity, but there is differences between insect species. So on the left, you can see Drosophila, where the placement of cell bodies can be in slightly different locations, or the soma, but the general architecture is conserved. And I think this is really exciting, the potential of making connectomes from other species and really using high resolution morphological data to understand how shape evolves throughout the tree of life. And this just summarizes the number of NSCs that we found in the different systems, and it differs, but not dramatically. So we continue to look for more. We're actually pretty close, we think, to identifying them all based on the symmetry between the two brains. But there's actually different classes of neuropeptides in ants, and actually probably the hardest part is finding antibodies that can identify all the different neuropeptides and to do validation studies on the ants, and also getting the ants. It's been really hard to get a permit to get the ants transferred to us. So we're also using, in addition to identifying synapses in the ant connectome. So this is an example of where there's a prediction made by the AA model, and then we have to go in and actually validate that and identify which synapses are real and which ones aren't. So it's a real iterative process where the company Zeta creates a model, and they send us the synapses. Then we have to go through like specific regions to actually validate. And so we've gone through several iterations, and it's improved quite a bit, quite significantly, I would say, since we started. And so these colored areas show where the predicted synapses are. So the pre-synapse would be the non-colored cell, and the post-synapse would be the colored region. But unfortunately, Zeta didn't have a model to predict the particular types of synapses that secrete neuropeptides. And this is our main focus in the lab, so we decided to initiate our own model. And so working with, in collaboration with Yervand Azanian's lab and Alexander Bates, we're developing our own EM model. It involves identifying, so most, we've known from many years of study that most dense core vesicles are around the soma, or the cell body of the neuron. And so this was really useful in being able to just segment the somas out and then compare them. And in doing that, we actually were able to predict and identify dense core vesicles throughout the neuron pretty successfully. So it used a sort of, I would say, what's become a standard UNet segmentation model to identify changes in opacity between the different pixels in the different EM sections. And then we also apply a segmentation model that's more of a standard algorithm. But so far, it's predicting with an accuracy of about 80%, which is pretty good. And so we continue to, since the fly actually has multiple connectomes, this has been really useful for validating the model and comparing them. And so we can look across all the populations of neurons, and we can see which neurons sort of stand out in terms of having more dense core vesicles versus less. And thankfully, the endocrine system definitely is producing a lot, the most, but also the motor neurons, which was a little bit more of a surprise for us. And it really is a really rich source of hypotheses for us to investigate further. And it's amazing the level of detail you can get. And because of that, to do the validation, it does require still a lot of man hours to go in and manually make sure that all these are true. So we have many undergrads that are helping us with this process. And more kind of full validation of what we expected, where we see the NCC has the highest density of discourse. So this is the region of the brain that actually secretes into the hemolymph. So that was exciting. So now I'm going to segue to a sort of a broader topic. This idea of using AI to connect biology using high dimensional data. And it really made me think of this really cool idea I learned about probably 20 years ago, the idea of consilience, this idea that if you have evidence from multiple different types of fields and experimental methodologies that converge on an answer, that tends to be a right answer. And this idea was actually developed by the famous philosopher and polymath William Whewell, who wrote the book, The Philosophy of the Inductive Sciences. He actually coined the term scientist. And he actually saw science as the collection of a bunch of facts and then sort of like putting them together and seeing if you could see patterns in them. He called this super-inducing. And when you learn about AI, you think, wow, that is AI, right? It seems to be the computational equivalent of this idea of the colligation of facts and finding patterns. And I actually was introduced to this idea with the incredible book by the incredible former entomologist, ant specialist, E.O. Wilson, who actually predicted that we would sort of take our understanding of complex biological systems to the next level by reassembling them in a mathematical model to form ensembles of that information. And this process has begun. So you may be familiar with the universal cell embedding model, where they're taking cross-species single-cell atlases, it's essentially as many as they can find, and combining them in a single embedding model. So this really heavily depended, it turned out, on the protein language models. So proteins have an incredible ability to relate function and sequence into a very compact embedding that allows you to actually predict many aspects of cellular function just from the sequence's structure. But these models, what sort of takes them beyond doing the traditional sequence homology is their ability to actually compare structure as well. But unfortunately, it's not been very successful for embedding arthropod single cell data. So it's for several reasons. One big one is the incredible imbalance in single cell data sets between vertebrates and essentially everything else. And I think maybe this has something connection to the plant biology, where we could definitely use a lot more data in regard in order to make a real accurate predictive sort of embedding that encompasses all cellular features in life. And then there's some major challenges doing this, in that you can only consider genes between species that have one-to-one orthologs. And often, there's a lot of loss of genes or expansion of genes that can make this really challenging. So you're limited to just a few genes to compare single cell data across. But fortunately, there's been some really cool and useful advances using supervised learning and deep learning that can compensate for this. One of the coolest is this SATURN method that uses metric learning or ability to compare one known annotated single cell data set with another annotated one using the similarity between protein language models. And with this, they actually found that you could overcome this orthology challenge using this new concept they call macrogenes. So they actually map each gene from each different species to a new AI-created feature called a macrogene. And the macrogene, you can have as many genes as you want connected to a given macrogene. So you don't have to lose things that don't have orthology. And because you're comparing structure as well as sequence, you actually can find functional similarities that you would not normally find with sequence alignment. And also new ways of integrating single cell data. I'm really impressed by the single cell variational inference method, which allows you to consider many more highly variable genes in your single cell data set. So in the past, you were limited to like 2,000 to 3,000 genes that you would then run PCA through. But with this method, you can actually consider 10,000. So this gives you a lot more potential genes to find similarities between. And then just to sort of finish it off, in the Drosophila field, in the arthropods, we have a unique chance to include incredible structural information from sequencing data, from fluorescence microscopy data that has been gathered for many decades. And there's this ongoing project that has been really successful called NeuroBridge that can actually connect the specific split GAL4 drivers, the genetic drivers that define neurons based on their morphology, to the EM connectome. And since we can also connect these drivers to the single cell data, this offers, I think, sort of the first possibility of connecting transcriptional data to something as highly dimensional as the connectome. So just in summary, a growing collection of these synaptic scale connectomes has revealed this deep structural homology of conserved neural circuits across the animal kingdom. AI is playing a pivotal role in actually making these models and annotating them. It's also in single cell analysis, combining supervised and deep learning methods is bringing us closer to being able to embed all cellular data into a single embedding. And I think the arthropod field has a unique ability with the amount of work that's done over the last few decades to actually combine ultra structural features on the scale of a whole organism to the transcriptome. So with that, thank you for watching my presentation. I would just say that so Meet is just starting his lab in Belgium, is looking for excited, interested people to work with. So please contact him if you have any interest in this. Thank you.
Thank you, Mr. Gruner, for a fantastic lecture. At this time, we will open the floor for questions from the audience. As a reminder, please place your questions in the chat. Our first question from Alan Wang. How do you deal with overlapping leaves? Hans, I think it's for you. Can the system automatically identify overlap and project and or exclude and or otherwise correct for that?
Yeah, thank you very much for the question. I would answer it in such a way that you, for sure, based on the like an outlier on the projected leaf area, you could identify overlapping or like connected components. And for sure, there are algorithms out there, like watershed algorithm, for example, to segment also overlapping or connected components. The study was done mainly in 2022, so there's also a huge jump in software development. So currently, this was a research project, and I would answer that way. Yeah.
Thank you, Hans. Next question is also for you, do you see a time where we move from organ on a chip being organized to being organ on a chip being AI silicon chip? I think it's for Matt.
Yeah, definitely. They're calling this the AI virtual cell, I think is the name I've heard. And the idea is, if we can, as much of the biology as we can virtualize, the quicker we can do, any testing all kinds of ideas on it. Essentially, running ablation studies on all the parameters of the embedding, that kind of thing. And I'm actually really fortunate to be near the Bay Area where a lot of this work is being started, and it's it's at a very nascent or new level, I would say, and it's really yet to show that it can really focus in on like better laboratory experiments, but the potential seems pretty big.
Thank you. At what temperature do you set the bench to prevent condensation within the Petri plates, Hans?
Yeah, that's also a very nice question. So, and I can answer briefly. So the room temperature was around 23, and then we made the culture cold feed that the water condensation will happen at the media level and not at the lid. And this was with a temperature gradient of around 1.5 degrees. This is already sufficient.
Thank you, Hans. Another question, Matt, for you. How do we scale up the generation of synaptic scale connectomics?
Yeah, so there's another, I would say, transformational technology they call expansion microscopy. So this allows us to you embed a tissue in this material that can evenly expand it in all directions. And then this allows us to use just fluorescence microscopy to do the same resolution of imaging that we're getting with EM data. And in fact, one of our big collaborators, Niels Reinhardt, is just starting a lab in Germany to use this technique on arthropods. So I think this will actually bring connectomes into the lab and throughout the world and allow a lot more experimentation and generating a lot more data. Because really, we just need more data. I mean, the AI is extremely data hungry.
Thank you, Matt. We will wrap up at this point. Thank you, Hans and Matt, for sharing your insights. And to everyone involved in this webinar organization from SIVB and CAST, at this time, I do want to remind you about our annual meeting. I hope to see you all in Reno, Nevada, June 6th to 10th. It is going to be an exciting event, once in a lifetime event, because we are celebrating our 80th anniversary. I hope you have a wonderful day ahead, and I look forward to seeing all of you at our next webinar, in addition to in Reno for our 80th anniversary celebration this June. Thank you all.